Before I have the opportunity to introduce today's speaker, I'd like to begin by acknowledging the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of the Kulin Nation, and pay my respects to the elders present, past and present, and the elders from other communities who may be here today. So it's with great pleasure today that I have the opportunity to introduce Dr. Jeremy Burrows, who is the Vice President and Head of Drug Discovery at the Medicines and Malaria venture. So Jeremy has 20 years of drug discovery experience. He is an inventor on over 25 patents and co-authored over 90 publications, many of these in the field of malaria. So Jeremy is an organic chemist by training, so we won't hold that against him, and attained his PhD at Oxford in 1996. A year later, he joined AstraZeneca in the UK as a medicinal chemist working on infection, cardiovascular and inflammation research, and some years later is seconded to the Swedish arm of AstraZeneca, where he led a program focused on Alzheimer's research. In 2008, he joined the Medicines and Malaria venture, where he was appointed head of drug discovery. So at the turn of the century, approximately $4 million of seed funding was provided by various funding organisations, and the Medicines and Malaria venture was launched as an innovative platform or collaborative model known as a product development partnership model. And this was in the field of antimalarial drug research. And it was really at this time that there was well over 1 million people dying from malaria each year, and the antimalarial clinical portfolio was totally devoid of any new clinical candidates coming through the system. And so it was a really dire situation at the time that the Medicines and Malaria venture was formed. So today, the Medicines and Malaria venture has over 35 partners worldwide and is almost solely responsible for populating the antimalarial clinical portfolio with numerous candidates to treat this devastating disease. So the Medicines and Malaria venture is now not only the world authority in developing or taking small molecule starting points into the clinic and to the marketplace, but is also leading the charge to eliminate malaria from various regions around the world through development of preventative treatment regimes. So I'm sure you'll agree with me after viewing Jeremy's seminar today that this product development partnership model that the Medicines and Malaria venture has in place has certainly been what I'd call an outstanding success. And so now I welcome Jeremy to the stage to present his seminar.
So it's really fantastic to be here in Melbourne with you all. And Brad, thank you for that very kind introduction. Now, there is a term called estimated net present value, which essentially means how much something will cost you today versus what you think you'll get back to it in the future. And this is something that businesses are concerned about when they make projections about their budgets and what they work on. Now, from a drug discovery perspective, it's clear that, well, in any business, in fact, you want to keep this estimated net present value to be negative for as little time as possible. You don't want to be working and burning money without any expectation of being able to recoup it. If you work in a disease which is actually has a commercial focus, like Alzheimer's disease, you can see that during the discovery and early clinical development phases, it's all in the red, but really by around about phase 2, you start to get into the positive. And this, of course, is because of the promise of riches that will come if ultimately you're successful. The big problem with respect to drug discovery and development for malaria and other similar diseases is that when you calculate these net present value, essentially you are in the red pretty much until you launch a medicine. In other words, no one in their right mind would go into antimalarial R&D to make money. You do it to save lives. Now, this effectively has led to market failure. What are the opportunities and what are the solutions to market failure? So as Brad said, one of those solutions is actually to come up with new ways of working, new models for R&D. And this led to MMV, Medicines for Malaria Venture, being founded at the end of the 90s with the mission to discover, develop, and deliver safe, effective antimalarials for the world's poor. We are a not-for-profit foundation, and as Brad mentioned, we're a product development partnership, and this term is really special for us because it emphasizes that we're not just interested in exciting, excellent science, but we want to deliver products. We want to deliver medicines to patients. And our goal is to cure and protect the vulnerable and help ultimately eradicate malaria. So who are we? Well, there we are. Roughly 80 people based in Geneva. We're like a virtual pharmaceutical company, if you like. Roughly half of us are scientists from industry. And on our own, we cannot do very much. And indeed, everything that we achieve has been managed through collaboration and partnership. And it's wonderful, to be here with friends and colleagues from the community. So a new model to do drug discovery is one aspect. MMV to coordinate the global effort. Another is obviously to thank the donors that we have, and obviously to hope that other donors will come onto the scene. And I just want to start off by saying a huge thank you to the folks that give MMV money, including the Australian government, the Bill and Melinda Gates Foundation. GHIT Fund from Japan has been extremely important in recent years, and there's a whole host of other governments and agencies. And then one of the opportunities that comes from working in a disease where the commercial pressures do not drive you towards secrecy is the opportunity for openness. And I guess this is a theme that I want to develop through the talk and really introduce you to several areas of openness using the definitions that I will take you through: open science, open innovation, open access, and open source. So what about where we are in the landscape in terms of combating malaria? So the first thing to say is that there's really good news from over the last 15 years. Huge progress has been made from the turn of the century when things looked very, very bleak. And since the peak, the number of cases and the number of deaths and the number of cases have come down significantly, essentially equating to a year-on-year decrease of between 2% and 4%. And this is really, really good news. And this is clearly due to the use of bed nets, but a substantial part is also to the use of chemotherapy. Something happened in 2007, which was very, very significant. Astonishing leadership from Bill and Melinda Gates, standing up, shocking the community, saying, "We believe in nothing less than eradication." Margaret Chan, head of the World Health Organization, came on board, daring the community to join in the fight. And in fact, the WHO have sort of built on the success that's been achieved in the last decade or so, and actually now set goals for where the community needs to be by 2030. And you can see that by 2030, the global malaria strategy has set the ambitious goal of a 90% reduction in cases and mortality. And in fact, the Gates Foundation have gone beyond that to actually start to look at a pre-elimination phase before ultimately eradication. Interestingly, if you do the maths as to what actually this means on a year-by-year basis, we're now looking at 16% annual decrease versus the 4% that I mentioned earlier. Clearly, there's a sense in which to achieve this, we are going to have to shift gear. This is not going to be feasible without a dramatic increase in resources across all of the activities that are necessary to deliver this outcome. So for malaria, in terms of chemotherapy and drugs, the standards of care are fixed dose combinations. So they're fixed dose for compliance and combinations to combat resistance. And the products that we have, they are all, in fact, have their origin in two natural products. So this is artemisinin, for which you two got the Nobel Prize, and this is quinine. And essentially, quinine then led on to compounds like mefloquine, also synthetic analogs, the 4-aminoquinolines, essentially was born out of the structure of quinine. And the great news is that these antimalarial combinations, they are really efficacious, they're well tolerated, they're stable in tropical conditions, which is a really important criterion for drugs for NTDs, and they're affordable. So a full treatment cost of Coartem, is around about 75 cents. Coartem D. So Coartem D is Coartem dispersible. It's a child-friendly, cherry-flavored version of artemethyl lumefantrine. This was launched by MMV and our partner Novartis in January 2009. And since then, it has been in 300 million patients. I mean, staggering numbers. So that's one child's life transformed every second since the drug was launched. So all of this is really, really good news. There's obviously bad news, too, about drug resistance. And this is a major challenge, not only because we are now seeing artemisinin resistance, this delayed parasite clearance time in the greater Mekong subregion, but also because of use of monotherapy of some of the drugs, but also because the artemisinins are starting to fail, we're seeing partner drug resistance, so piperacin resistance really becoming a problem, for example. So we absolutely need new drug combinations comprised of molecules which have different mechanisms of resistance and action. And then when it comes to Plasmodium vivax, which has this dormant form that sits in the liver, the hypnozoite, at the moment we have one drug, which is primaquine, an 8-aminoquinoline, which is a 14-day therapy. You can't give it to patients who have low-active glucose-6-phosphate dehydrogenase because of the risk of hemolytic anemia. Also has GI side effects, too. This is a problem. From a compliance perspective, the great news is that we have low single dose even, tafenoquine, which is in phase 3 with our partner GSK, and this is looking very, very nice from the phase 2 study, and we're waiting to see what the outcome of that is. It is still an 8-aminoquinoline, though. It still has the risk of anemia in G6PD deficient individuals. So we need new drugs to kill hypnozoites or activate hypnozoites that don't have that risk if we're going to be serious about eliminating Plasmodium vivax. So this is how the portfolio looks at the moment, as of sort of halfway through the year. And you can see that MMV, with our partners, we've delivered a number of products. Interestingly, everything to the left of this line, these are new compounds, new mechanisms of action, which really have come since around about 2007. There is a large research portfolio, and we believe, again, this is absolutely critical to continually fuel this pipeline of drugs going into preclinical human volunteers and so on, because there is major attrition in drug discovery, and particularly then when you think that we are going to be combining drugs, the risk for failure is even greater. Just to show you the extent to which we've made progress, even since sort of 2008, let alone 2000, as Brad mentioned, this is how the portfolio looked 8 years ago. So there's been a substantial level of investment, but that investment has really had impact. So when you do a drug discovery project, it's really important to be very clear about what you need to deliver when you start your project. And this is really beginning with the end in mind. It focuses on the idea of a target product profile, a TPP. Now, Pedro Alonso led a considerable activity looking at the research agenda for malaria eradication, and they published a series of papers in PLOS. We sort of built on that, really on the idea of two major TPPs, one for treatment and one for protection. And in the ideal case of each of these, it's actually a single intervention. And so, for instance, for treatment, we're talking about ideally delivering a drug combination which will be a SIRCAP, a single exposure radical cure and prophylaxis. In other words, that combination medicine would have aspects of fast clearance of parasitemia, ideally post-treatment prophylaxis, killing gametocytes to block transmission, and killing or activating hypnozoites. So ultimately, those parasites are killed. Now, from a drug discovery perspective, where we actually work on an individual molecule rather than on combinations, it's really important to understand how a molecule itself can relate ultimately to the product. And so this is where we come up with the idea of target candidate profiles, where we define the criteria associated with each of these attributes that will then deliver the ultimate combination medicine. So TCP1, for instance, defines the criteria associated with a molecule delivering fast clearance of parasites in patients. Or TCP3b is compounds, criteria for compounds killing gametocytes that then affect transmission to the mosquito. So essentially kind of how it works is that when we screen, or when we run a project and we have front-runner compounds from within a project, we actually test those compounds in assays that we've developed with our partners around the world, from either academia or from industry, against a range of assays which essentially give us information about activity on the parasite life cycle at all of those points where we believe drug intervention is feasible. And so, for instance, if we screen against a Vivax liver stage assay and we find compounds that kill hypnozoites, that can then link back specifically to a TCP and then link back to a TPP. So there's a link between the life cycle fingerprint, the phenotype that we get from a chemical series, and actually, ultimately, what we want to deliver. Of course, the assays that I showed, these are all non-clinical assays, assays in vitro and animal model assays. But it's important, once you deliver a clinical candidate and you actually move into human studies and patient studies, that we have protocols that allow us to measure quickly and effectively to confirm those activities in patients. And so, for instance, we have clinical relapse protocols. We have experimental blood stage challenge studies. In fact, we do these in Brisbane. And I will say a little bit more about those later in the talk. Now, interestingly, as we've worked on chemical series, we've actually, over time, found information with respect to the mechanisms of action of how these compound series, operate. And essentially, if you take the phenotype of a particular compound series working on a particular target, and you superimpose it onto this sort of Venn diagram shown, where this is supposed to represent compounds active against liver stages, this should really be a gametocyte. It's compounds active on gametocytes and compounds active on asexuals. You can begin to see that specific mechanisms of action, specific targets, have a particular phenotype. And this is very, very much what we see. And interestingly, again, this actually shows that an individual target can link to a target candidate profile, can then link to a target product profile. What this then leads to, again, once we have a screen, which often is a phenotypic screen, the first thing that we do is we test against these phenotypic assays around the life cycle. We get this life cycle fingerprint. We know which part of this Venn diagram a particular compound activity sits. With our partners, again, we've set up biochemical or wholesale assays to help deconvolute. And so, for instance, if a compound hits asexual blood stages and late-stage gametocytes, one of the key immediate things that we think about is this an ATP4 inhibitor? And for instance, we send this to Kieran Kirk at ANU, who runs sodium homeostasis assays, and we have many, many mutants that we can look at also. And this helps us to, rapidly get information, at least where we have an assay and we have, knowledge of a particular mechanism. And we use that for either prioritization or deprioritization as necessary. So when we do projects focused on asexual blood stages, what are the critical assays that we use? Well, the workhorse assay, and certainly for screening, are the growth inhibition assays in vitro. Another assay that's very, very important for us, because it gives us definition as to actually how the compound affects parasite viability. We get these very nice kill curves. It's something we call the parasite reduction rate assay, which is an assay that our colleagues GSK at Tres Cantos set up a few years ago, and gives information in vitro which is just unparalleled. And you just cannot get it from standard growth inhibition assays. Once we have compounds that are acting where we understand the growth inhibition, we understand the kill rate in vitro, we have good pharmacokinetics and physical properties, we actually start to do in vivo studies, and we actually use a falciparum, well, a skid mouse, where you engraft human erythrocytes infected with falciparum malaria. And what's really nice about this is that effectively it sort of mirrors what you're going to be seeing clinically. You allow parasitemia to rise, you add your compound, and you obviously would then like to see rapid parasite clearance. And then to confirm activity, we actually go into a human challenge model. So this is where we infect volunteers with blood stage malaria. You see the periodicity of infection. This is all an asymptomatic level, I should add. And then at a certain point, the new chemical entity is given. We get a very rapid readout of the effect of that compound, and clearly would add the standard of care were it not to work. But in fact, every compound so far we've taken into the challenge model in Brisbane, has given nice parasite clearance. What about Vivax? Well, huge progress has been made with Vivax liver stages in recent years, and it's really quite astonishing, actually. And we've been working with Clements Cochrane in the BPRC on cynomology. But for instance, Dennis Kyle, who's based in the University of South Florida, working with Francois Noston at SMRU, Mysot in Thailand, he's now effectively established a 384-well liver stage assay using primary human hepatocytes, which are essentially spatially confined in a specially molded 384-well plate. And so we now actually have the ability to do this. Whilst, it's important to say that access to sporozoites is obviously still, the actual key issue, because we have to go to endemic regions, we have to feed mosquitoes on patient blood, dissect out, and then ultimately, generate the assays. In terms of blocking transmission, the assays that we're really after, and the approach that we have, is to target the stage 5 gametocytes, indeed, ideally, the entire gametocyte population. Because it's the stage 5 that infect. And targeting the vector stages on their own, whilst a bonus activity, if you like, is not a strategy in its own right, because you do not know when a mosquito is going to bite you. So we've, with our partners, again, set up assays based on gametocytes and based on gamete formation. And then the gold standard is really the standard membrane feeding assay. And indeed, this has now even moved to the position of being able to look at the effect of compounds on transmission, at least to the mosquito, in humans in a challenge model at QIMR. And I was in Brisbane last week, and Catherine has very nice, a pot with 30 mosquitoes, which are fed on patients who are full of gametocytes, and then you can look to see whether you could potentially dose a compound and look to see whether that actually blocks infection. And then for chemoprotection, so chemoprotection is really in the context of the end game scenario where elimination of malaria is actually becoming a reality. Malaria has come down and even been eliminated in certain regions. Quite understandably then, the immunity of a population will decrease, an entire population will be vulnerable, the vector most likely will still be there. So all you need is for someone to come in full of gametocytes, and there's risks of outbreak. And so we need safe chemoprotectants to sustain the gains that we will have got and to protect those vulnerable populations from the breakouts. And we have in vitro, in vivo, and from the vaccine world, there are now sporozoite challenge models in humans too. So I mentioned that I was going to weave in this theme of openness. The first term I want to define is really open science. And we believe at MMV very much about making our data available as early as we possibly can in order to aid the community. And this really means getting it out into the literature or sticking data into public databases. And in fact, with our partners, I mean, GSK, Novartis, and St. Jude had amazing, courageous leadership to make all of their high-throughput screening data available in 2010. And we've worked with Kemble to actually pull together all the data that's publicly available in a one-stop shop in Kemble. And we also push for very early sort of publications of warts and all papers, essentially, on our clinical candidates that are moving forward. Because this data is really, really valuable for the whole community in terms of developing the next generation. But in fact, the main way in which MNV operates is something that I'd like to define as really being open innovation. And open innovation is really where two or more partners come together with a very defined goal to deliver something, which in our case is obviously a candidate drug and then ultimately a medicine. But through that collaboration, everything is open and everything is shared. And in fact, within the MNV model, I showed the slide with the life cycle with all the assays around them. Anybody joining the MNV family, if you like, then immediately has access within a confidential membrane to all of these partners, and we have the ability to then facilitate sharing and openness of working. So how do we actually find new candidate drugs? Well, the first route is simply to look at the drugs which are out there and what can they teach us about how they're operating to then bring forward a new entity. And so the first example really is from Jonathan Benestrom. So this is Jonathan, who was the PI for this project from the University of Nebraska. And Jonathan really, spotted that the artemisinins have this endoperoxide, and that this could be mimicked synthetically by an ozonide, a trioxylane, which is this kind of motif here. Which is, I mean, I'm a chemist, I'm a medicinal chemist, and this is a pretty scary motif from a medicinal chemistry perspective. But, Jonathan went with the data. He didn't go with any prejudice. Made a variety of compounds, and interestingly, this compound, very simple compound, extremely potent against malaria. Trouble is, this compound is brick dust, totally insoluble. So what the team had to do is they had to find parts of this molecule where they could build in hydrophilicity to improve the solubility and the properties. The first candidate that came from that work, so you can see that they were able to build off this cyclohexyl ring, sticking on this amide and this amine. This was actually the first candidate, OZ277. In fact, this is now a marketed drug in India as part of a combination with piperacin called Cinrium. And I'll say a little bit more about that in a moment. But in fact, the team went on to optimize OZ277 further because it was actually found that in patients, the pharmacokinetics of this compound was substantially worse than they had been in volunteers. And so the team then actually took that information and focused on improving the stability of these compounds in infected blood. And essentially found that this sort of syn-phenyl ring protected the peroxide, leading to compounds with very nice stability in infected blood. And indeed, this compound, OZ439, is currently in a phase 2 single-dose clinical study with ferroquin. So OZ439, as I mentioned, the next step is actually to take it in an in vivo animal model. This is our SCID model, and you can see that this gives very rapid parasite clearance from a single dose. In fact, just to say a bit more about how we use this animal model data and actually, how we're thinking about it, typically what we do is we do a dose response and we get a concentration dose dependency. We relate that concentration to a particular effect. So this is the PK, this is the PD, the pharmacodynamics. And then what you can do is you can take that information and mathematically model it. And essentially then see if you can simulate the data. And so this is the simulation at the bottom right here. You can see the dots are the actual values, the dotted lines are the simulations. And you can see the simulation using the standard model works very well in this case. When you extract the parameters out of a model, you actually can then very effectively see how the compound is effectively working in that mouse. And so, for example, you have the green concentration of compound over time. The model gives us the growth rate of parasites within the in vivo system. It gives us something called the MPC, the minimum parasiticidal concentration, the concentration above which you have maximum parasite killing or clearance. And the MIC, the minimum inhibitory concentration, the concentration above which you inhibit growth. And And you can see that the parasitemia decreases at a maximum rate until the concentration drops below the MPC, and then growth only starts again once you go below the MIC. And the idea is that we can do the similar modeling on compounds tested in the human challenge model, tested in phase 2A, and then relate all of those parameters to see exactly how we're translating from the non-clinical situation, even in vitro, in vivo, challenge model, phase 2. So coming back to the story of OZ439, so I mentioned OZ277, which was the first candidate. Just to show you, this is a plot of compound concentration over time to really emphasize the improvements in pharmacokinetics of these particular compounds. Here's DHA in green. Many of you will be aware that DHA, dihydroartemisinin, isn't around for very long once you dose it. OZ277, the first synthetic peroxide candidate, and volunteers actually had much improved PK. You can see this blue curve here. But we then went into patients. And it's a very interesting example of the disease affecting the pharmacokinetics. And in fact, the pharmacokinetics were significantly worse. OZ439 designed to be good both in volunteers and in patients. You can see very nice consistent exposure, way over the MPC, giving us the real hope of efficacy with a single dose. The next question is obviously to see, does this lead, do these concentrations that we see lead to efficacy? And so we've actually done dose responses in Australian volunteers in the human challenge model. And actually the beauty of dosing, if you like, suboptimally, allows you to see the recrudescence and get very, very good markers of these parameters that I mentioned earlier. And then clearly to actually test in a phase 2A where you actually go into a patient population. Typically we like to go into patients who are not fully immune, or into African patients, for example, because we want to understand exactly what the contribution our compounds are making to parasite clearance versus immunity. So for instance, for OZ439 against both falciparum and Vivax, we have very nice clearance data and parasite reduction rates. So with OZ439, and with any compound that gets to this point, the journey really is to ask the question, what are you going to combine it with? Because antimalarial medicines will be combinations. And it's a real question, it's a real tension between, do you go with something that is already marketed, that we know a lot about, where we understand the weaknesses and the strengths? Do you wait for something beautiful to come along? And in fact, a lot is talked about actually the, how do you make those decisions around what you combine with? One of the very practical points to mention is simply that you have a limited choice of really what is at a similar phase. We have various matrices and decision-making tools for helping us prioritize combinations. And in fact, for OZ439, it's currently undergoing a single-dose combination study with ferroquine, which is a 4-aminoquinoline. Other ways in which you can find new candidate drugs for malaria is essentially the reductionist approach, which essentially is target-based screening that effectively is the modus operandi of the pharmaceutical industry. And this is where, from genomics, you have understanding of a particular target, you validate it, you set up an assay, and you run a screen, you find compounds, and then you take those forward. And we've done this with a few cases. I guess until fairly recently, there haven't been a lot of good validated targets for malaria, but I'm very pleased that that's changing. The most advanced example is actually the dihydroorotate dehydrogenase project, which was led by Meg Phillips out of the University of Texas Southwestern. So she ran a high-throughput screen and actually found a very potent compound right from the start. This compound actually has a number of liabilities, not least the fact that this beta-amino naphthalene is one of the most mutagenic compounds known to man. Also, having a poor pharmacokinetic profile, changes were made to the aniline. This started to give compounds which actually were active in vivo, even though you'd lost a little bit of potency. The crystal structure actually then came online at that point, and it was clear that it was possible to build off into this direction. And SF5 as a sort of an optimal group to give potency and very low clearance led to DSM-265. So DSM-265 was then taken into a phase 1 study. And so here you can see the pharmacokinetics from different doses of DSM-265 in humans. And a number of things to say here is, first of all, it was well tolerated between quite a substantial dose range. In fact, the PK looks really nice, this lovely sort of linear PK. From all the predictions of what we had estimated based on killing concentrations, we would anticipate that this is the concentration that you would need to be above in order to then have efficacy. So predictions were able to be made about what the efficacious dose would be. So we then looked in a human challenge model, and the first thing to start is to look actually at suboptimal dosing. So this is in fact with 150 milligram dose, and you can see that the drug works. Then the exciting thing is obviously to then take that information, or both the PK and the challenge model data, which supports this idea that 300, 400 mg single dose would have efficacy in patients. Moving to a phase 2A clinical study in Peru. Here's the data in the 10 patients that were studied in the first sort of cohort, and you can see, in fact, out of those 10 patients, 9 out of 10, in fact, were cured, and this is actually going out to day 28, which is way better actually than we ever anticipated. And in fact, there was just one failure. So DSM-265 is now in the position of thinking, which partner do we go with, and then moving the compound forward. So another way in which we can find hits is actually to ask the parasite. And in this case, we're simply wanting to get hold of as much chemical diversity as possible, to screen them against the parasite, to find hits, to move these towards candidates, essentially optimizing in a phenotypic manner, in the process, identifying mutants through which we then get information about modes of action. And really, through this exercise, we've now screened over 7 million compounds with our partners, and we've got a range of hits. I mean, this looks a big number, but in fact, once you start to whittle it down based on duplication of targets or duplication of scaffolds or unpleasant chemistry, it actually starts to come down to low numbers. So we still believe it's important to access new, exciting chemical diversity. In many cases, this has actually been not only productive, but quick, actually getting into man from screening relatively speedily. And in terms of identifying new targets, there are a whole host of things that we've pulled out, which effectively were not necessarily the most important targets that the community were thinking about at the time. So the first example was really led by Thierry Diagana from Novartis Institute for Tropical Disease. So this was a major collaboration with Novartis. They screened 2 million compounds and picked out a singleton hit, this spironolone. And from a medicinal chemistry perspective, this is it's a potent compound. This compound was actually active in vivo, in the Bergier model, and so this was also something that helped them choose the particular derivative. Things that they wanted to change, they wanted to reduce the lipophilicity of this molecule because this is high. And so bromine to chlorine is a standard change, truncating the ring and getting clarity on the enantioselectivity here. Very pleasingly, actually gave an even more potent compound. A great way to go. But clearly the intrinsic clearance is high. And so they needed to actually improve the intrinsic clearance, find compounds with better pharmacokinetics. And so blocking the metabolism in this ring actually improved potency further, dealt with the stability issue, and gave the first candidate. And this, was a whole phenotypic-based approach which only led to an understanding of the mechanism of action right at the very, very end. In fact, after the publication of the science paper, when, in terms of really understanding, more about it, when Kieran Kirk saw the science paper and then phoned up Thierry, and that led to a discussion where all of Kieran's work over many decades was able to bring to bear on this particular series. And this series inhibits ATP4, a very exciting new target for malaria. And basically, the parasite clearance associated with even low single dose from this compound is quite astonishing. So parasite clearance in 12 hours. And to put that in context, that's substantially faster than the artemisinins, which would be 24 hours. So another example is from Kip Guy at St. Jude, who did a high-throughput screen, finding this particular compound after a range of optimization, really focused around potency, physical properties, and pharmacokinetics. Changes to this aniline, this unit here, and the nitrogen substituent led to the identification of SJ733. This has actually also then turned out to be an ATP4 inhibitor through a completely different scaffold. Has a very interesting, safety profile. And excitingly, this is a project which, with Assay and GHIT, is now currently in phase 1, and actually studies are ongoing in Brisbane with James McCarthy with this agent right now. Another example of a candidate, this time a kinase. This is the first malaria kinase inhibitor, led from a project led by Kelly Shabali in the University of Cape Town. So Kelly worked on a series of minor pyridines that was identified from a biofocus screen by Vicky Avery in Ascites. He found these hits that have some clearly non-optimal functionality here, which they immediately sort of focused on, giving some very nice profiles. But there were still optimization that was necessary. And again, when you show it like this, it makes it all look so easy and so simple, but there's huge amounts of compounds and effort and understanding that goes into each of these arrows. A simple change of methoxy to CF3 has led to a compound that's extremely stable, very, very potent. And this really was able to, this project was able to move forward. Early exemplars were in vivo active. We found essentially that the mechanism of action through both pull downs and resistance studies was this new kinase PFPI4K. And astonishingly, the selectivity is outstanding, much better than ever we could have rationally designed. And this particular inhibiting this pathway actually leads to relatively slow killing. So you need compounds that have very, very good pharmacokinetics. And indeed, taking this compound into human volunteers demonstrates very, very nice pharmacokinetics in humans, and has also clinically validated the target. Another example from the University of Dundee, and this is a lovely example where actually they screened a very, very small library, around about 3,000 or 4,000 compounds. They found a hit, and being good medicinal chemists, they wanted to get rid of the lipophilicity, which they did. Again, solubility, and it was something that they wanted to improve, so they changed this pyridine to this amine, even at the expense of potency. And in fact, it's really important that physical properties are not sacrificed, on the altar of potency. Based on other SAR they had, they were then able to bring everything together to identify DDD498, which is an exquisitely potent compound, across the entire life cycle. Also identifying a new mode of action through resistance work, which is elongation factor 2, and was published in Nature last year. Another example from the group of AstraZeneca. So this is with AstraZeneca India in Bangalore. We did a high-throughput screen of their company collection, 500,000 compounds at the time. Actually, this particular hit was not super potent, and actually has a number of liabilities. But the team did a tremendous job of very systematically identifying those issues and working through them. And there were a range of things, as I say, that they had to fix. This was the first compound that they sort of considered as the lead, and further optimizing that has led to MMV253, which is now moving forward in preclinical development as a completely new mechanism of action. We know the mechanism of resistance, but we don't really understand how the compound works. And I want to sort of close by just weaving in a few other themes around openness. First is open access. And so open access is actually where someone deliberately makes data or compounds or protein available with intent. And this has got to be actively communicated and managed. And we've very deliberately had an activity with something called the Malaria box. And this really came about because our partners published all of this data. And people said, this is really great, but ideally we'd like compounds. Can you give us 20,000 compounds? And we said no. But what we can do is we can be very pragmatic and find out how many of those are commercially available, how many would we need to actually pull together to form a reasonably fluent set that would capture that diversity. And we chose 400. And we established this box. And it's now been made available to over 260 groups worldwide. Interestingly, people have used the malaria box for different things. Some have screened it against malaria, their favorite target, or whatever, special assay. But in fact, a number have used it to look for actives against other pathogens, for example, or even screening against cancer cell lines. And all of the data, is now, all the data that we're aware of is pulled together in Campbell. And we recently had a kind of like a CERN-type publication where we contacted all the recipients of the boxes and said, send us your data, and we slung it all together in a massive, massive spreadsheet, which obviously allowed those who'd got negative data on all of the compounds to add their data set in, but nevertheless giving information, to the community. And there are partners who have found hits on helminths or cryptosporidium, for instance, that have then led to actually funded projects with various foundations. This led on to something called the pathogen box. So we're very grateful to the Bill and Melinda Gates Foundation for giving us a grant for this. And essentially the idea here was that we would pull together a set that would not just be focused on malaria. We'd use all the learnings that we had from the malaria box, but this wouldn't just be focused on malaria. It would comprise compounds active against one of all of these pathogens. And these compounds would be, selected by a group such that they would be reasonable chemical start points for programs. This was launched at the end of December last year, and since then, they've been flying off the shelves. So over 130 boxes have been shipped, worldwide. And in the box, roughly 100, a quarter of the box is tuberculosis, a quarter is malaria. Canidiplastids is the next biggest one, and so on. And if you're interested in the pathogen box, you just simply need to go to the pathogen box website and, click, fill in, tick a few boxes, and they'll send it to you for free. Finally, open source. So this is the idea where you actually work in a way that information is then made available to the community, such that the community can then engage fully, so that the community helps to improve what was originally there. And we've been very fortunate in this respect to work with Matt Todd, who's a professor at the University of Sydney and a real champion of the open source way of working. And this has really been an experiment for us in terms of a model of drug discovery that can actually bring in a wider community, with the idea that significantly larger input could actually really move a project forward. And there's some very nice work that's been achieved and recently published, and the team are currently working on a ATP4 series that came out of a Pfizer high-throughput screen and is looking interesting. We've benefited in what we call this MMV open space, actually with huge support from both academics and folks in industry who've come to MMV on sabbatical. And so I just wanted to mention this, and if this is a call that you think, then come and talk to me. Finally, just to wrap up the talk, I just wanted to end with just a few points of learnings and some challenges. So I guess the first thing to say is that in terms of really having success with the drug discovery cascade, you have to have good assays. And these have got to be robust and cheap and high throughput. And in fact, I think it's the miniaturization of assays and the reduction of cost per well in assays that have really allowed us to access and screen diversity. We've also had a framework, a legal framework, which essentially has really facilitated collaboration with people. You can have all the best people and the best scientists and whatever, but unless you can have the legal frameworks that allow this to happen, it's a disaster. And I can talk more about that if you're interested. We've prioritized hits based on life cycle stages and rates of killing, because we are interested not just in control, but in elimination and eradication. And where our projects have moved forward most quickly it's where the hit itself has been of the highest quality. Clearly, you always need to focus on good medicinal chemistry. And knowing the biological target can be really critical to navigate through safety issues. And there are projects, such as the DHODH project I mentioned, where we would not have got to where we are now if we had not understood what the target was and what the activity on mouse and rat and dog and whatever DHOD was. And with new mechanisms of action, the critical thing is to get these through into man as quickly as possible to then validate and demonstrate really how they operate. And governance is critical. We have an independent, expert scientific advisory committee, which is very candid in its feedback, and that really helps us, to prioritize. And actually, there's a lot of success that we've had where we've basically been extremely lucky, and I think this is just something that we've got to humbly, accept that we can't rationalize our way through all of these points. In terms of challenges, there are certain mechanisms of action which we repeatedly find. And this means that we need to deconvolute, and that's a lot of effort, and it's a lot of work to do that sort of thing. And clearly, this whole why is it that we find so many compounds that are active on particular targets? I mean, I'd love to hear your view. I mean, is it some of these issues here? We don't know. I mentioned before, whilst we have many hits from phenotypic screening, once you actually start to pare the list down based on quality and prioritization, you actually end up with very few. And so there is a real need and a real question mark about where do we find the next level of diversity to screen. And in fact, in that particular regard, we've actually got historical compounds where you can actually come back to look at these compounds and look at them in a different way through a different screen as technology improves. And that can really rediscover interesting options. And I do believe that there is going to be a renaissance in target-based drug discovery for malaria. I mean, it's very, very clear just how the community has, responded and the tools that are available to do this. It's very exciting. And I really focused on asexual blood stages. But for those of you who are very observant, there's a critical question around screening for gametocytes and hypnozoites. Just to end then, this is really an image of how MMV operates. We receive donor funds and in-kind contributions, from partners, intellectual contributions, monetary in-kind contributions. And then with our partners, both from academia and from industry, the fruit of our work together are the new medicines for malaria. And with that, I'll leave you with a quote, and thank you for your kind attention.
Thank you, Jeremy, for an incredibly insightful talk. I think we've got a little bit of time for a few questions.
So if you get a super drug or a bunch of super drugs, then one of the biggest problems, I guess, would be in countries where there isn't really the social organisation to deliver it in an effective way. Thinking of Africa, places where as much just being shot as dying.
So, I mean, that's a great point. So the question, if you didn't hear it, was really, even if you have a great drug, what about actually the issue of getting that to patients in some of the most difficult areas within the world? Interestingly, this exact point was made by Kevin Marsh at the ICTMM meeting in Brisbane last week. Because in fact, the situation now is that we have good artemisinin in combination therapies which are working very well in Africa. I think the figure that he quoted was something like 20% of people who need to get those drugs actually get them. So there clearly is an access issue. And even with the right tools, unless that is actually fixed, how are we going to actually combat, control, let alone elimination? I mean, you're absolutely right. I mean, MMV has an access team, essentially, that's really focused on ensuring that we have approvals within countries, and we unblock as many of the limiting factors as possible. But there are other structural issues and that need to be overcome, and strengthening the internal national health malaria programs, for instance, But it's a great point.
I was wondering if concentrating on those hits that have great efficacy right from the start, you actually set yourself up to detect the same target or similar targets repeatedly, and that the other hits that don't look so promising initially and maybe haven't got the same chance of resulting in an efficacious drug would target potentially other.
Yeah, so it's a great point. So the question was basically the way in which we prioritize, does that bias what we find, and is that one of the reasons? I think if we take, for instance, our test cascade, asexual blood stage, Bergii liver stage on actives, and then confirmation, say, on gametocytes, that's been a very good route to find PI4K inhibitors, and I do wonder whether that test cascade biases, in that way, and the similarity of that enzyme across those species, for instance, is particularly, similar. In essence, it doesn't really matter if what you say is true, because we should prioritize on those things which are most effective to begin with. But clearly, once we've actually prosecuted those, it's not that we've forgotten all of these things back here. We do absolutely come back, and mine them.
Thank you for a really exciting and inspiring talk, a new way of doing business, as your slide says. I noticed that MMB was actually allowing products to be used in research on other infectious agents. But do you think, if you were planning a second adventure like MMB, do you think it's important to tackle, for example, antibiotic resistance in the same way as you've done for malaria? How many of these do you think the world, these organizations, do you think the world can actually sustain at the moment?
So I guess
that means the goodwill of companies and academia.
Yeah. So how many product development partnerships are really needed? And so I guess one of MMV's strengths and individual product development partnership strengths is the fact that they focus. We are just simply focused on small molecules for malaria, and that is really our goal. We're about delivering those medicines for patients. Antimicrobial resistance, Chagas disease, TB, a whole great list of things that are major problems. Many of these do actually, there are product development partnerships that exist which are very focused on tackling those issues. DNDi, the Global Alliance for Tuberculosis, drug discovery and development, for example. Antimicrobial resistance is an interesting one because right now there isn't a single global point where people would go in order to tackle that. And I think basically that's going to work itself out in the coming months and years, because there are a number of organizations which are stepping up and saying we will do similar types of activity in AMR. But as I say, it just needs the landscape needs to settle down and for us to be clear of the way forward.
I'll take one last question, Jeremy.
You mentioned the dream of eradication of malaria, but isn't that, despite a couple of things you said, isn't it likely that you'll actually have to get rid of the mosquito vectors to have a hope of doing that?
So if I gave the impression that eradication was possible simply through chemotherapy, then I apologize, because I don't believe that. You absolutely need to tackle the vector. I guess the worrying thing at the moment is that a lot of the insecticides in Africa are failing. The pyrethroids are failing. But there is a product development partnership called IVCC based out of Liverpool, which is focused on the next generation of insecticides. And they have a range of interesting insecticides that combat the existing resistance, which will come online in the next few years. But Jeremy, I absolutely agree with you. We have to tackle the vector too.
Okay, I think we'll close this forum, and I think you'll agree with me that the Medicines Malaria Venture have made some remarkable progress in such a short period of time, which really shows that this model that the Medicines Malaria Venture have has really worked quite outstandingly. And join me in thanking Jeremy for a wonderful talk.